Good morning, everyone, and welcome to session 2, module 2 on signal detection and signal management, part of the African Union Smart Safety Surveillance Capacity Strengthening Training. I'm Mick Foy from the MHRA, and today we're going to be joined by 3 members of our team at the MHRA, Anastasia St. John and Samantha Myers, who are signal assessors in the vigilance team. Will be taking you through an introduction to signal detection. And then Catherine Tregano, a scientific assessor in our benefit risk management group, will be taking you through signal assessment with some particular considerations on vaccines and medicines in pregnancy. The next slide, please. So as a reminder to colleagues who have joined before, or an introduction to those that this may be the first session you've used, with our use of Zoom functions, you will be muted during the course of the webinar. So we ask you to use the chat function if there are issues around any technical issues or functionality of the running of the webinar, but please use the Q&A function if you have a question for any of the speakers today, and we'll pick those questions up at the Q&A session towards the end. We've seen a number of people in previous sessions use the raise hand functionality. Unfortunately, the way we've kind of locked down the sessions, we can't respond to raised hands, so we always ask you to use that Q&A box. As a reminder, these sessions are recorded, and we will be making these available as we've done with all the previous sessions. So if you have colleagues that are unable to make the session, you can always point them in the direction of the recordings. Next slide, please. So again, we have a large number of colleagues joining us. I'm told we expect around 200 or so this morning from across Ethiopia, Ghana, Nigeria and South Africa, but also other organizations, as you can see on the slide here. And you're all very welcome indeed. So this is module 2 of our capacity strengthening training program, which will run today and tomorrow. We'll conclude module 2 on signal detection and signal management, and then in March we'll be moving into issues around benefit risk assessment and risk communication to follow. So today's session 2, as I've said, we'll look at that introduction to signal management and some specific considerations in signal detection. And as always, the opportunity for Q&A at the end. So please use that Q&A box and keep your questions coming in. Our aims today, or throughout the module, last week we covered that overview of causality assessment and how that is carried out, looking at different models. And now we're moving on to an understanding of signal detection, and then tomorrow we will move in to provide an overview of how signal management meetings are conducted and some of the concepts that are used at MHRA that hopefully you could adopt. So without further ado, I will hand over to, is it Anastasia or Sam picking up first?
Thanks, Mick. Yes, it's me. I'll kick things off, and thank you for that introduction, and good morning, everyone. So myself and my colleague Samantha Myers are both signal assessors of the signal management team, and together we will give you an introduction to signal management at the MHRA, as Mick has previously stated. So just to provide an overview, in this presentation we'll be covering what constitutes a signal and the sources of signals, and then signal management processes at the MHRA, which includes how signal detection, validation, assessment and prioritization are carried out. Next slide, please. So what is an adverse drug reaction, or an ADR as we will refer to its abbreviation from now on? So as you may well be aware, an ADR is a response to a medicinal product which is noxious or harmful and unintended. These adverse drug reactions may come about from the use of a medicinal product within the terms of its marketing authorization or outside of these licensing terms, for example, in scenarios of off-label use, as it's called, for use of the medicine outside of the license indication, or in the instances of medication errors such as overdose or drug misuse. It's important to note that ADRs reported to us via the MHRA's yellow card system or scheme, or other routes, are not a confirmation that the reported reactions are definitively associated with the drug. They may well be just a temporal in their association or a mere coincidence. We encourage patients and healthcare professionals to submit reports of ADRs on the basis of a suspicion. As a consequence, this generates a hypothesis for which investigation and examination of the evidence is required to determine the causality of the ADR to the drug. And this is the objective of causality assessment, which has previously been discussed by Claire Davies in the previous session, and will be discussed in more detail with regards to signal assessment later on. Next slide, please. So what is a signal? A signal is the combination of a drug and an adverse drug reaction event that is a possible safety issue of the drug. This drug and ADR event combination is abbreviated to DEC, so drug event combination. In the textbook definition, a signal is considered to be an alert from any available data source that a drug may be associated with a previously unrecognized hazard, or that a known hazard is quantitatively or qualitatively different from existing expectations. So in theory, signals are not just novel safety concerns that arise. They can also include alignment issues for the product information between different products of the same substance, or a change in the existing warnings for certain medicines use, given new information that has come to light, or the need to create additional monitoring requirements in light of new evidence from the post-marketing experience. Next slide, please. So the process of signal management in pharmacovigilance is defined as the set of activities which aim to determine whether there are new risks associated with a particular drug, or whether risks associated with a particular drug have changed. These activities include signal detection, signal assessment, signal validation, and signal prioritization. Next slide, please. So how are these signal management activities conducted within the MHRA's process? To provide a brief initial overview, firstly, potential signals originate from various sources, either as ADR reports or from other possible sources. Then signal detection and initial signal assessment is carried out on the drug event combinations. Then any potential signals are presented at the weekly signal meeting. Then if the meeting decides further action is needed, the signal is validated and further assessment is required for the review at the signal management review meeting. This is where an initial action is agreed along with a timeline for action, and this is the final signal prioritization step. Next slide, please. The MHRA detects signals for a number of sources in order to maximize our detection scope. These include spontaneous data arising from either our yellow card submissions directly from healthcare professionals and patients, or indirectly from industry partners. We may receive notification of a signal received directly from the pharmaceutical companies themselves, or ones identified in published literature and a number of other studies. Signals may also arise from other regulatory authorities through data sharing or via notification of procedural outcomes from the European Medicines Agency, for example. We additionally can identify signals from general queries we receive from healthcare professionals and patients alike. Usually, it is from spontaneous data that we identify most of our signals, but we must recognize the importance of other sources of information further to the yellow card scheme, as the scheme itself has its innate limitations that are to be expected. Next slide, please. When adverse drug reaction reports are received, they are uploaded into our case management system called Sentinel, where a team of assessors make sure the details of the case are entered correctly using medical coding conventions in order for accurate signal detection to take place. For example, here you can see that the signal report is as it appears on Sentinel in our case management system, from which a signal concerning warfarin, an anticoagulant-related nephropathy, was recently identified from spontaneous yellow card data. So as you can see from Sentinel, that in this one report, that a number of suspect medications were reported, including warfarin, tamoxifen, and tacrolimus, and a number of adverse drug reactions were classified in response to the patient initiating these medications. Next slide, please. Once cases are committed to our Sentinel system, they are automatically added to the program that we use for signal detection called Empirica. When the previously discussed ADR report was committed to the signal management database Empirica, each reaction, as you can see, is now presented as a single drug event combination. So here we have the warfarin and anticoagulant-related nephropathy as one row, and separate to this, there is a row for warfarin and drug interaction, and another for warfarin and acute kidney injury, and so on. Whilst we know the context of the original yellow card ADR report saw all of these reactions occurring with the 3 reported suspect medications, it's important to be able to look at the individual drug event combinations in order for signal detection to be carried out, with the context of the original case being manually looked at by the assessor. Next slide, please. So the next step in the overall process is signal detection and initial assessment. Once a signal is detected, the objective of signal assessment is to examine the evidence for a causal association between an adverse reaction and a suspected medicinal product, to quantify this association, preferably in absolute terms, and to identify the need for additional data collection or for any regulatory actions. It consists of a thorough pharmacological, medical, and epidemiological assessment of all of the information available on the signal of interest. Next slide, please. Our signal detection process scrutinizes ADR reports for potential hazards. Every new ADR report may represent a new signal. So when carried out traditionally, this is considered as looking for needles in a haystack. During routine detection, a weekly review of all fatal reports and reports deemed serious are assessed as a priority. Due to the large volume of reports received, the MHRA uses tools to help facilitate this decision-making in order to streamline the process and efficiently pick up on any signals without the need to review every single case initially. As briefly touched upon, spontaneous reports are entered onto the case management system that we use called Sentinel on a daily basis as they are received. They are then transferred to Empirica, the program we use for our signal detection activities. Through this program, signal statistical scores are automatically computed every week for reports received, and signals of potential interest are flagged for assessment based on predetermined criteria. As mentioned by Claire Davies in the previous session, at the MHRA we use the EBGMs as the statistical method to indicate a measure of disproportionality. This is automatically calculated for us via the software we use, and we don't manually calculate the score for each drug event combination. We have 2 teams to assess signals at the MHRA. One team assesses signals for established medicines, and the other team assesses signals for black triangle or additional monitoring drugs, as different criteria applies for these 2 sets. The team assessing black triangle or additional monitoring drugs have more experience and are specialists in their field. Next slide, please. So to further aid our signal detection, the available data is automatically subgrouped into categories such as vaccines and non-vaccines, as the data output for each of these requires a different approach and background for statistical comparators. Statistical filtering is applied to the subgroups to further help with identifying new signals. This filtering is based against results of statistical disproportionality, commonly where the number of observed events is greater than what is expected. For both established medicines and additional monitoring or black triangle medicines, it is estimated that around 85% of drug event combinations received are filtered out, as they do not meet one or more of the statistical criteria, which is vital for our resource capabilities. Additionally, a rule-based approach is applied to further narrow the search. If an allocated signal row contains one of these rules or meets the statistical filtering criteria, it alerts the assessor to manually investigate this, allowing us to assess signals in a multifaceted fashion. Next slide, please. So the traditional method of signal detection involved reviewing every new ADR report. However, at the MHRA, we have a large database where over 1,000 reports are received each week, so manual review is not possible. So we use statistical methods to automate signal detection to identify drug event combinations that are disproportionately present in our database. It's a systematic and practical means of screening large data sets, and it has positive contributions to public health by identifying potential safety issues sooner and more accurately. However, it does have some disadvantages. Pharmacovigilance systems may generate errant signals that turn out to be false alarms. This can occur because other factors confound the apparent relationship. Inadequate analysis could also conceal a true risk signal that might have been evident with more careful adjustment. Next slide, please. So Empirica will automatically flag the following by screening ADR case folders for established medicines. And the criteria include serious drug event combinations, where the statistic EBGM is greater than or equal to 2.5, or for the lower threshold, the EBO5, the score must be greater than or equal to 1.8 and have more than 3 cases. The criteria also include drug event combinations which are defined as serious by measure and or the SIAMS definition. So just to provide additional information on this, the serious criteria for ADR reporting, this was determined by a working group of the Council for International Organizations of Medical Sciences, or SIAMS, and are defined as 6 possible categories, which are documented on the yellow card. So reporters can select one or more of the following criteria by ticking the appropriate box in the yellow card. So the criteria include whether the patient died due to the reaction, whether the reaction was life-threatening, or if it resulted in hospitalization or prolonged inpatient hospitalization, if the reaction was a congenital abnormality, or if the reaction involved persistent or significant disability or incapacity, or if the reaction was deemed to be serious by the reporter themselves. So Empirica will also automatically flag to us all unlisted drug event combinations and listed drug event combinations that meet the EBGM disproportionality criteria, where a specific change in the reporting frequency is detected. And this is based on a comparison of the proportion of reports received in the last quarter. Empirica will also flag reactions from fatal reports, reactions from reports involving children, reactions from parent or child reports, including occurrences of spontaneous abortion, reports for alert terms, which are medical conditions of interest, which are more likely to be caused by medicinal products. This list is loosely based on the important medical alerts list from the European Medicines Agency and was confirmed internally by a team of specialized medical assessors. Assessors for Black Triangle or additional monitoring drugs review all UK reports for their products, except for non-serious child cases. However, this is due to be updated to ensure all UK cases are reviewed. Assessors are expected to present all serious, unlisted reactions, excluding first cases with no UK cases, all serious reactions from fatal reports, and all unlisted alert term reactions at our weekly signal detection meeting. And I'll now hand it over to my colleague Samantha for the next slides. Thank you.
Thanks, Anastasia. So hi everyone, my name is Sam and I'm going to talk you through the rest of our presentation. So there are 4 main statistical methods used for signal detection processes, and this slide quickly highlights which regulatory authorities use what. All methods identify drug event combinations that are disproportionately present in the databases, and each method has their own benefits and disadvantages. As previously mentioned, the EBGM, or Empirical Bayes Geometric Mean, which is used predominantly by the MHRA, is a statistical measure of disproportionality comparing the observed and expected reporting frequency. The determination of expected reporting frequency assumes complete independence of cases associated with either a drug or an event. So if you imagine a database of 100 cases where 20 cases are with the drug paracetamol and 10 cases are with the adverse drug reaction rhabdomyolysis, the expected reporting frequency would be 20 over 100, which is the probability of the case being related to paracetamol, times 10 over 100, which is the probability of the ADR being rhabdomyolysis, times 100, which is the size of the database. So you would expect there to be 2 cases in the database where paracetamol has been reported with rhabdomyolysis as the adverse drug reaction. If there were actually 8 cases with paracetamol reported with rhabdomyolysis, then the relative reporting ratio, which is the observed over the expected, would be 8 over 2, which would be 4, and the EBGM would be slightly less than 4, depending on the exact shrinkage applied to the model, which reduces the confidence intervals around the EBGM and accounts for doing multiple comparisons. For example, the chance of finding a statistically significant result purely by chance, which would be a false positive, as well as instability created by differing market shares within the database. The value can be skewed with very rare adverse drug reaction terms in the database, resulting in a high EBGM for very few case reports. However, this occurrence is normally quite obvious. For example, an EBGM of 33 with 2 reports would flag this. Next slide, please. Each row represents a drug event combination that meets the predefined criteria, and each column represents the associated statistical measurement. It allows for a high-level, quick overview of the number of reports and gives us easy access to view these case details to obtain the context of each case. This helps in determining the presence of a signal. Reaction terms can either be assessed as isolated drug event combinations or as group standard MEDRA queries or fatal preferred terms. Next slide, please. As many of you will be aware, the MHRA used MEDRA to code our ADR reports as well as for identifying alert terms. Our automated signal detection process generates potential signals using a drug and a MEDRA preferred term. This is because many lower level terms map to the same preferred term. Similar preferred terms are grouped into standard MEDRA queries or SMQs, and these aid our signal detection. SMQs are groupings of MEDRA reaction terms that relate to a defined medical condition or area of interest. As you can see to the right of the screen, an example of an SMQ for lactic acidosis contains a number of MEDRA preferred terms, either with narrow scope, which are terms highly likely to represent the condition of interest, or with broad scope, which is more likely to pick up possible cases that may not represent the exact condition, so has a higher sensitivity. SMQs can prove useful in early signal detection of signals when we have a limited number of ADR reports. Next slide, please. Fatal special PT groups are another useful grouping technique that detects an increase in the expected number of fatal reports for a particular drug substance and are generated as part of our automated signal detection, appearing as its own separate row for signal evaluation by assessors. Fatal PTs must meet the statistical criteria on the slide, as discussed by Anastasia earlier, and are reviewed at least every 6 months. Upon this analysis, the fatal PT data is run to determine if there are any large unlisted clusters of fatal events that we would need to be concerned about. Next slide, please. To give an overview, whilst the MHRA uses Empirica as a tool to aid in signal detection, the automation for ease of signal recognition is transferable across platforms. If you could click next, please. Each ADR report from the UK is reviewed weekly for signals originating from the UK. Next, please. We then can see the reason this drug event combination has flagged for further assessment based on the criteria it has met. Next and next again, please. All the statistical calculations and specific cases are available along the row for each drug event combination present within the committed cases. Next and next again, please. And finally, similar terms should be looked at to gauge the possible extent of the signal, as this gives broader understanding. For example, If a signal of suicidal ideation was being investigated, similar terms, such as depression or mood alterations, will need to be reviewed in conjunction, as may be supportive of the original signal, or as depicted here, similar cases may be retrieved by using the higher level term function. Next slide, please. Once a drug event combination is deemed to be a possible signal after meeting the predefined criteria, further manual assessment is carried out. How we assess signals generally follows a similar pattern, and we will recap on this causality assessment, which was previously covered by Claire in the previous session. The biological plausibility is assessed and might be predicted from the known pharmacology of a medicine. This can be supplemented with reviewing other medications within the same drug class. Timing of the onset of the reaction is important to look at if available. For instance, most of the time, anaphylaxis is an almost immediate reaction experienced. And so, if the time to onset is not would be as expected, such as days or weeks post-administration, this would be carefully considered and suggestive that true anaphylaxis may not have actually occurred. Adverse drug reactions can be related to too high or too low dosing of the suspect medications. This can be identified as potentially under or overdosing and can be considered a medication error, as would fall outside the remit of the licensed dose regimen. This is particularly important when over or underdoses are intentional, or if they occur as a result of human error or device issues, as these may act as justification for the reactions experienced. Additionally, if symptoms appear when the medication is withdrawn, it suggests that there is potential to be associated with the medicine, known as a positive D challenge. If the symptoms recur upon reintroduction of the medication, this is a much stronger indicator of causality. A suspected reaction may also be a manifestation of an underlying illness or associated with another medicine being taken by the patient concurrently. Or additionally, it may be entirely coincidental. Whilst we are reliant on the statistical calculations, when disproportionality is met, it does not always indicate an unrecognized signal, and all factors need to be considered alongside the statistical measure. Medications may behave differently under different conditions, and the consideration of the possibility of pharmacokinetic and pharmacodynamic interactions with other medicines or food and drink or medicinal devices should be undertaken. Next slide, please. To recap and provide indications of timelines, signals are allocated to assessors on a Monday morning. At the signal detection and initial signal assessment step, a review is conducted of all allocated drug event combinations in Empirica by scientists from Monday through to Wednesday. Potential signals of interest are then discussed in our weekly signal meetings on a Thursday, as this allows assessors enough time to review all possible signals for that week. Also on a Thursday, a separate pregnancy signal detection meeting is held where every report relating to pregnancy or lactation is discussed. But my colleague Catherine will talk you through this in more detail later on. Next slide, please. So what potential signals might be discussed at the signal detection meeting? Assessors may choose to present potential signals for these reasons. The signal isn't labelled or listed in the product specifications. A report is fatal. A novel drug interaction that isn't labelled and we have more than one report of this interaction. All reports involving patients under the age of 18, so any child-related cases. All parent and child reports that result from exposure during pregnancy or breastfeeding, including spontaneous abortion. Alert terms that are not labelled, and any signal that is of concern and does not meet the above criteria. The most important criteria here is to bring anything of concern. The signal meeting can then discuss and decide on any concerns for further review or action. Even if an adverse event is labelled in the product information, an increase in frequency, increased or changed incidence in a specific patient demographic, or increase in severity may require further action. Next slide, please. During the signal meeting, each assessor presents their signal for that week. Each signal is presented and it's followed by group discussion on the signal. They will present their signal, why they're bringing it to the meeting, the available evidence, and their recommendations for any further action. So who is at the signal meeting? We have a team of multidisciplinary experts with knowledge across different areas of medicines attending the weekly signal meetings. This is to ensure we're able to reasonably comment on all signals brought to the meeting. The meeting is a platform for discussion, where people can bring any other business here for advice from the meeting as well as signals from the routine detection. If the meeting decides further action is needed, the signal is taken to the Signal Management Review meeting. Signals going to the Signal Management Review meeting have signal assessment case holders created for them. This is to enable adequate documentation and audit of the signal. An alternative route at this point is that it might be that at the time of presenting the signal, the meeting requires additional information or evidence from which to base the decision on for the next steps to take. In this scenario, the drug event combination will be recorded on the minutes, with the view to be brought back and presented again with the additional information. For drug event combinations that do not present the potential to be a signal or a safety concern and are thus not presented at the meeting, comments are to be left on each of these drug event combination rows in Empirica. This, in addition to the signal assessment case folders made for validated signals, are our primary method of documentation and audit of signal assessments. Next slide, please. So, empirical comments contribute to our audit trails. There is a set of standard comments such as listed in the product information, event is ongoing, event is being monitored, and so on and so forth. There is also the opportunity to leave a detailed comment to free type as extensively as necessary for the assessment that was conducted. These comments can be viewed by other signal assessors should the drug event combination return another week, allowing a cumulative tracking process. Detailed comments should be added where analysis has been completed in order to prevent duplication of work and to justify the actions that has currently been taken. This allows for a regulatory memory. Next slide, please. So once it is determined for signals raised, the signal detection meeting and the pregnancy signal meeting that further action is warranted, this signal is now validated and is to be presented at the Signal Management Review meeting. It is at the stage of Signal Management Review meeting that the appropriate regulatory action is decided after further assessment and discussion. The assessment case folder is created in order to document the progress and outcomes of the signal. Next slide, please. Prior to the Signal Management Review meeting, further assessment of the signal is required, with the need for all possible data sources and evidence to be gathered and evaluated. This is communicated firstly through a written scientific report called the New Signal Report, which is circulated prior to the meeting and then briefly presented verbally during the meeting. Within the New Signal Report, the assessors need to review all cases we have on our database, including all UK cases reporting any similar measure terms, or even cases for drugs within the same class of medicines. All other possible data sources need to be consulted to provide additional evidence to consider evidence such as literature, periodic safety update reports, the product information, or any other available clinical guidance. The meeting will then evaluate the strength of the evidence and discuss the most appropriate action to take to bring about optimal patient safety outcomes. Next slide, please. It is at the Signal Management Review meeting that the signal prioritization step occurs. A signal is assigned a priority based on a variety of factors, and this priority will dictate the timelines required for the action to be carried out. Lily Wells will provide further details on this meeting and the prioritization of signals in the next training session. Thank you for your time and for listening, and I'll now pass you over to Catherine, who's going to take you through the vaccines and pregnancy aspects of signal detection.
Oh, thank you. Sorry, I can't seem to start my video. Just a moment.
There you are.
Oh, okay, right. Thank you. Sorry, I just couldn't see myself there. Okay, thank you. So my name's Catherine Trigano, and as Mick said at the beginning, I'm a benefit risk assessor at the MHRA in the Benefit Risk Management Group. And I have been working in the unit which
primarily deals with vaccines and anti-infective agents. So today I'm going to follow on from presentations given by my colleagues Jolyn Chewy in Module 1. She spoke to you about considerations for ADR reporting and with vaccines, and a lot of the points and the considerations that she raised at that time are similar to those which you use when you are conducting signal detection for vaccines. And also my colleague Claire Davies presented last week on causality assessment. And again, those principles you would be using for vaccine assessment and assessing signals in pregnancy and breastfeeding. So today we'll just talk a little bit more about how there are slightly different considerations that you would have to take into account when you're looking at signal ADR data and also ADRs in pregnant and breastfeeding women. Okay, and I'll touch a little bit on MHRA processes as well for the pregnancy and breastfeeding ADR data. Okay, can I have the next slide, please? Right, so you just heard from my colleagues Anastasia and Sam about the signal detection processes that we use at the MHRA. And for vaccines, just to say, to start off with, that this is similar to drugs. So we have the yellow card reports coming in, and these go onto the ADR database that we have. And then we have the signal detection software, Empirical, which takes the data and then does the statistical calculations to present us with the listings that we get every week, highlighting the drug event or the vaccine event combinations that we should be looking at based on our criteria. And then these are allocated out for assessor review. So as my colleagues mentioned, we have the same signal thresholds that we would use. So with the EBGM of 2.5 and whether there's more than 3 cases. However, the data for vaccines are kept, the background data is based on vaccine background data, just because you tend to get a very different reporting profile for vaccines. So lots of things like injection site reactions and reactions to do with reactogenicity, which is slightly different for medicines. So you might have some masking of signals going on. All the new vaccines that we have are monitored by benefit risk assessors that tend to look at vaccines, just because then they have the specific knowledge about the slight differences with vaccine safety monitoring. And with the signal management process, again, this is the same as presented by Sam and Anastasia just now. So the same processes as for the medicines. So we have the weekly signal meeting, and then anything which is of concern would be carried forward to the signal management review meeting. And if it's considered that we need more advice, because we've got an idea about what we might want to do, but we need some advice from experts in the field, then we can take the signal for expert advice to back up our decisions, or to get more of an idea of what might be good to do. Next slide, please. Okay, so when you're doing signal detection with vaccines, what would you want to focus on? Obviously, for new vaccines, so novel vaccines like the COVID-19 vaccines that we're using in the UK now, these are the first 2 that we've really used on a large scale. So the mRNA vaccine, the Pfizer vaccine, and the Oxford AstraZeneca vaccine is based on an adenovirus. And while this kind of concept has been used in an Ebola vaccine, obviously we don't use this vaccine in the UK. So the aim, ideally, COVID aside, would be to review all the reports. However, we have had a lot of reports for COVID vaccines, so we have had to slightly modify our approach, and I will talk a little bit more about this in a moment. So of course, looking at all the serious ADRs and particularly the adverse events of special interest, and my colleague Jo Lin spoke about these kind of events in her presentation on vaccine ADR reporting in module 1. And just as a reminder, these there's information on adverse events of special interest that places like organisations like the Brighton Collaboration have lists of what they consider to be these AESIs, so things which you might possibly expect to occur following vaccination, albeit very rarely based on experience with previous vaccinations. So things like Guillain-Barré syndrome and other peripheral neuropathies, things like transverse myelitis and encephalitis, also things like stroke, cerebral and other cerebrovascular events, autoimmune events, things like idiopathic thrombocytopenic purpura, and then other ADRs related to exposure in pregnancy, which we'll talk about shortly as well. The ASIs would probably be vaccine specific. So when you're doing your vaccine signal detection, it depends on the vaccine that you're looking at. And hopefully you would already have a good idea, especially for a new vaccine, because of the pre-planning that you've done. And we will talk more about this in module 3, probably when we're talking about risk management. But yeah, so you would have an idea of the ASIs you would be looking at. So you're more directed towards particular terms of interest. And obviously you want to look at any fatal adverse drug reaction, because as we know, vaccines are given to healthy people to prevent disease. So you have a very different tolerance of the risk-benefit balance, and any kind of fatal outcome after a vaccination, obviously the temporal association is there, and that can cause a lot of alarm. So we'd be looking at those. Pregnant women, obviously this population is always of interest when we're doing signal detection, because these patients aren't involved in clinical trials. And there is a concern about the health of the mother, and also the effect on the unborn child. ADR suggestive of medication errors. With vaccinations, this may be something like if the vaccine is being given in the wrong place. So with vaccines given in the arm, injectable vaccines, sometimes if they're given too high up, you can get, an effect on the functionality of the arm. So if we see things like that coming through the data, then we need to investigate to see whether that's just a localized issue or whether it's something which is more widespread. And this is when you need to come to interact with the public health authorities, who have more of a reach in terms of reaching the vaccinators to issue communications to correct people's technique. Also, lack of efficacy. When we think about signal detection, we think about looking at safety signals. However, for vaccines, if you've got a vaccine which is, there's an indication that the disease is occurring in people that you've vaccinated to protect them against that disease, say meningitis or pneumococcal disease, then cases that are reporting disease after vaccination need to be investigated to work out whether has the patient had the full course of immunisation. Has there been some other factor which has affected the fact that the patient has gone on to develop the disease? And again, this is really just a first step to doing the signal assessment. You'd need to then go on to collaborate again with the relevant public health authorities to try and capture data on disease surveillance to see whether there's a shift in the dominant strains of a disease, So for example, pneumococcal disease, there is a lot of different strains, and the vaccines are carefully constructed to make sure that they provide protection against the most dominant, the most prevalent circulating strains, but that can change over time. So this is where disease surveillance from the public health authorities, that information comes in to play. And also, if you're looking at a live vaccine, so the MMR vaccine or the varicella zoster vaccine or a yellow fever vaccine, there are specific considerations, specific events that you would be looking at. So for example, a suggestion there's a reversion to virulence. So is the vaccine causing disease in individuals, and also secondary transmission. So this is a concern when you have people that are vaccinated possibly transmitting the disease onto immunocompromised contacts. Okay, next slide, please. So the challenges in vaccine signal detection, similar to the points raised by Jo Lin in the ADR reporting on vaccines presentation. So often given to a large percentage of the population, sometimes a specific percentage of the population, so be it elderly people or specifically children, or with the case of the HPV vaccine, given, although now in the UK this is given to boys as well, when the programme first started, this was only given to adolescent females. So you've got the potential for a large number of adverse drug reaction reports, and these you would expect perhaps to get a lot of reports of the reactogenicity. So the things that we know about, the things that we've seen in clinical trials, so injection site reactions, headache, fever, the flu-like symptoms, these kind of things. And if you've got a lot of these kind of reports, it can make it quite difficult to actually find the signals that are of real concern. However, this is not to say that having large numbers of cases of reactogenicity isn't of concern, because if it indicates that there needs to be better communication through the vaccines product information on what to expect following vaccination, then that's a way of looking at the data, working out what it's telling you. Are there some other aspects of the reactogenic events which need to be communicated? But in general, it can, if you're saying, okay, I have a new vaccine, I want to look at all the cases, but you're getting thousands of cases a week, then that makes it quite difficult, because obviously you only have a defined number of people probably. Obviously very rare and serious and fatal events will happen following vaccination. This is inevitable, and because the vaccine is just a one-off acute exposure, you can't stop it. So there's concern there. So this is where with vaccines, the signal detection with the ADR data is kind of the first stage of the signal assessment process. And it's really important to have other data to supplement the ADR data and to put it into context. So things like rates of disease, so incidence rates of your adverse events of special interests, and also details on exposure data. Do you know how many people are being vaccinated? If you've got 10 fatal cases that were reported in a day following vaccination, yes, this sounds concerning, but if you've vaccinated 500,000 people that day, and they're all elderly patients, then that puts it into context a bit more. Other challenges, so vaccines are biological products. There's no generic vaccine. So with a medicine, you might say paracetamol. We've got 50 different products of paracetamol, but we know where you are with that. But with vaccines, it's not the same. There can be different product variability for different flu vaccines. For example, we have several different flu vaccines that are used on any given flu season. So issues raised may not be just to do with the flu vaccine. So sometimes with when you're looking at the data in Empirical, it's quite tricky to work out if you haven't got product specific information. So somebody's just reported flu vaccine, you don't know which flu vaccine it is. That can make it quite challenging. And this is why we do our best to encourage brand name reporting, and also reporting of batch numbers, because within an issue may occur with a particular batch of a product, not even, you know, to do with the whole product itself. So this can be something which, yes, presents a challenge when you're doing signal detection and you think, oh, there's been some serious events with this vaccine. However, I don't know which batch it's to do with. Yeah, that can be a challenge. Also establishing the nature of the signal, whether it's something which is to do with the product or to do with the program. Is it to do with perhaps a breakdown in the cold chain somewhere? We may not have this information. Or it could just be an isolated issue. If you may have a cluster of reports with a particular vaccine and think, oh dear, that's concerning, because we've had lots of cases of infants spitting up a rotavirus vaccine after it being given. However, when you look at the data, they've all come from the same reporter, and perhaps it's to do with the way that the person was giving the vaccine. They weren't doing it in the way that they've been trained to do, which minimised the likelihood of the baby spitting up the vaccine. And also whether a vaccine signal may not be to do with safety, it could be to do with quality, which could have an impact on safety downstream. But it may be just to do with some manufacturing aspect to do with the vaccine. And this is where contact with the companies, and we have a defective medicines group, the MHRA, so we can liaise with them and they can liaise with the company to do investigations into the manufacturing data to see whether something has gone wrong along the processing chain somewhere. Okay, next slide, please. So I already mentioned the signal detection using ADR data. This is really just the starting point. When you're considering vaccines, you really need a more proactive approach, especially when you're faced with introducing a new vaccine, a new immunisation programme across your country. So the approach we've taken at the MHRA is a lot more proactive. So before the vaccine is deployed in the country, we undertake work to look at background incidence rates for the key adverse events of special interest that we're interested in for that vaccine. And that depends on the type of vaccine. Is it a live vaccine? Is it an adjuvanted vaccine? Is it being, which kind of disease is it being given for? Is it being given to a particular subset of the population? And for this, we look at either published literature or more often in the UK, we look at healthcare databases. So we have the Clinical Practice Research Database in the UK, which has records of primary care data and also mortality data. And this is important from the point of view of when you have these fatal cases which are reported following vaccination, because if you know, if you have details on mortality data, so how many people die of X age group, in a particular given time, then you can use this to put the numbers into context. And this enables something that we use called observed expected analyses, where you already have information on the number of cases for a particular ADR you might expect, based on the number of vaccines given and what would normally happen in the unvaccinated population. And then you can use your ADR data to put together and give you an idea of the signal. So whether is it happening more often than you would expect or not? And I think my colleague Craig will talk more about this in the epidemiology presentation in module 3. Okay, so also it's really important to have up-to-date vaccine exposure data and ideally age and gender stratified as well. And we're lucky in the UK that we are able to get this data quite quickly. And considering whether any more active surveillance would be feasible, so perhaps hospital sentinel sites, something like that, to supplement your spontaneous kind of passive ADR reporting. And also conducting ecological studies to look at the incidence of these adverse events of interest over time. So whether you're seeing any peaks which coincide with the implementation of the vaccination programme. Next slide, please. So I'll just run through some of the options that are available when you're considering vaccine signal assessment, signal management. So for a new risk, again, this is similar with medicines. You can update the product information or consider some kind of active communication to alert healthcare professionals and the public. However, there are considerations which need to go alongside this. And I think in module 3, again, you will have a presentation on effective communication, effective risk communication. If you have detected a systematic medication error, considering how to communicate the correct use of this. So just updating the product information may not be enough, because people who are very familiar with giving the vaccine are not going to be looking at the product information all the time. So there may be a consideration about how you reach the people vaccinating and how to do a more active communication. Again, as I mentioned, if you think there's a suspected quality defect with the vaccine, then the manufacturer needs to look into this, because they will have the data on the vaccine. And in the case of vaccine failure, I mentioned already, getting in touch with your public health bodies that are responsible for disease surveillance. If you're not sure, there's a signal and you think it could be a causal relationship between the vaccine and the event, it may be that you need to consider a study which specifically uses a hypothesis to specifically look at the association, because it's difficult to work this out just by looking at ADR data because of all the biases and the limitations. And you also may need to seek expert advice on proposed actions. And this is the case for vaccines or medicines or anything really. You know, we have ideas about what to do, but expert advice is always key. But this really depends on the nature of the signal. Sometimes you need to take immediate action. And then it may be that later on down the line, you want to consider seeking expert advice on other kind of risk minimization measures. Okay, next slide, please. So with the COVID-19 vaccines, I'll talk quite briefly about this. Obviously, I said, ideally with these new vaccines, we would want to be taking a real close look at all of the data that comes through when we're doing our signal detection. And we do have enhanced frequency of signal meetings. So whereas normally we have one main signal meeting a week, followed by the signal management review meeting, for the COVID vaccines, we have been doing several meetings a week, just because of the volume of data which is coming in and the need to have more of a kind of real-time approach to the data. And we have a dedicated multidisciplinary team. So we have pharmacovigilance assessors, so signal assessors like Sam and Anastasia, and then benefit risk assessors such as myself, and then my colleagues Claire and Jolynn from previous sessions. We have a dedicated expert advisory group because of the nature, the speed at which things are moving with these vaccines. We need a group that is kind of readily set up with the relevant experts in immunology and various aspects of public health to advise on any issues which arise. And we have really close linkages as well with our communications team in case an issue arises which needs communication, to help us work out the best way of issuing that message without causing alarm. Obviously, we don't want to cause unnecessary alarm, which may damage the vaccination programs, not just in this country, but globally. And we also need to have close links with the relevant stakeholders, so the pharmaceutical companies and also the public health bodies. Okay, next slide, please. Right, okay, I will talk a little bit about how we handle signal detection in pregnancy and breastfeeding. So again, we have the same mode of reporting. We have yellow cards which come in, which indicate an exposure during pregnancy or during breastfeeding, and these are subject to the same processing. However, all cases concerning drug exposure during pregnancy, either via because the mother's taken the drug or because the father's taken the drug, and there's a suspicion that there's an effect on the unborn baby because of that exposure via the father, or also any cases which suggestive of an adverse drug reaction because an infant has been breastfed and their mother took the medication. So all of these cases are identified. All of these cases are reviewed by a specific team of assessors on a weekly basis, and there is a weekly meeting which is specific to the pregnancy signals, and all of the reports are discussed here. So there's no distinction on the EBGM criteria, the disproportionality scores. However, this is generally manageable because we don't generally get a very large volume of these data. And any new signals of concern are then taken forward to be discussed at the signal management review meeting, which Sam was just talking about. And we also have a specific expert advisory group, the Medicines for Women's Health, EAG, and we can seek advice from them, and that's made up of experts in obstetrics and gynecology and pediatric health. Okay, next slide, please. So the considerations for signal assessment in cases involving pregnancy and breastfeeding. So again, similar, you would take a similar approach as for any medicine or for vaccines in terms of the causality assessment. However, if you're seeing a new congenital defect, which is unrecognised, then you would consider the patterns of outcomes. So not just looking at that specific event. So if it was a ventricular septal defect or a cleft palate or some kind of limb deformity, you would look further afield to look at how these kind of events tend to be reported with other defects. And also look for cases of spontaneous abortion with that medicine, or termination of pregnancy if we have this data, because these can also be suggestive of an effect on the fetus. Again, considering the potential confounders relevant here. So, and this really depends on the information you have on the yellow card, as it may not be, you may not have very much information, and this with yellow card data tends to be the case. So what are the underlying medical, what are the underlying conditions of the mother? So what's she taking the drug for? If it's a vaccine, it's slightly more complicated because there could be any number of underlying conditions. Is there any information on the previous pregnancy outcomes? But this typically requires follow-up with the reporter. And this can be quite difficult because if someone's just experienced an adverse pregnancy outcome and they've taken a drug, they may well be quite distressed. So it's sometimes difficult to get this information that you need. If you see a recognised defect or adverse pregnancy outcome with a drug that's a known teratogen, so for example, valproate is very well known, and some of the other anti-epileptic medications are known to cause congenital defects. Although it's known, you may want to consider whether there's a suggestion that the existing measures in place to prevent these kind of things happening, so for example, the pregnancy prevention programs, whether there's some new aspect which means that this isn't being followed. Okay, next slide, please. Okay, so this link here is I just added this in case anybody wants to go and have a look at the COVID-19 surveillance strategy that we have in the UK. I can always provide some other links to the other data that we're publishing for the COVID vaccine data, if that would be helpful. But other than that, I will thank you for your attention. And then I think we've got time for some questions now. Thank you.
Thank you very much, Catherine. And also thanks again to Sam and Anastasia. A real tour of how signal detection is done there. And I think the focus on vaccines there, Catherine, is very important to this audience. We have spoken previously about special considerations for, as you mentioned with Jolene, around ADR collection, but I think the signal detection aspects are really key as countries are now starting to roll out vaccines and looking at their immunization programs and how surveillance is actually going to be working. We've had a couple of questions come through the Q&A box. I would encourage people, if you've got questions, to put them in now so we can pick them up. I think this first one came in as Sam was talking, I think, around, is there I think the nub of it is there an assessment template for a signal assessor? Do we work to a template when we bring signals back to our meetings? I don't know if Sam or Anastasia want to pick that up.
Yeah, I can give that one an answer. So it's important to consider that all signals are different and we'll have different aspects to each of them, but there are common factors that we would consider as signal assessors across all signals when evaluating them. This will include looking at the usage of the medication. For instance, is it widely used within the population, and therefore could this reaction impact a large number of people? Equally, it's also necessary to consider the proportion of the adverse reaction within the usage, as if we had 10 reports for an event if a patient usage was of 1 million compared to 10 reports of an event in patient usage of 1,000, then we'd have to treat those with different priorities. Other factors we consider include the seriousness of the event. For instance, is this event a severely debilitating event, or can it result in hospitalization or permanent reduction in quality of life, compared to reactions that are transient and will disappear quickly or not have a large relative impact on the patient's lives? This also has to be considered. We additionally place emphasis on the strength of the evidence that we have. If the statistical measures are raised and we have other evidence away from spontaneous data, such as support from the literature or biological plausibility, where mechanism of actions would be suggestive that the event is plausible, this would be considered good evidence and we would be more likely to want to take actions. Obviously, there are issues with this factor, as can only really be applied to well-established medicines with multiple drugs within the same class to allow this comparison and view of mechanism of action. It would be difficult to apply the same standards to new black triangle or first-in-class drugs, and there's likely to be less information readily available for our assessments. The format of assessment that is taken by assessors will depend on the scenario and the context. So whilst we try and evaluate certain factors, all factors may not be applicable for each signal. I didn't know if Anastasia or Lily, you may want to add anything at all.
Yeah, I guess just to reiterate that the format that your signal assessment takes is based on the scenario that you find yourself in with the potential drug event combination and the context, and then also using your own judgment to kind of decide and steer which way and which direction your assessment will go in. So it's hard to, there's no one size fits all sort of format or kind of structure of how to do an assessment. It all depends on what's in front of you in terms of that safety signal, what the drug is, what the reactions are, what the patient population is. There are so many things to consider. So that's why this whole thing can't be automated, and it requires signal assessors like ourselves. And as much as we use, statistical methods to aid that prioritization, it always comes down to that judgment, that scientific judgment that you have to make when you are presented with all the evidence and what you see in front of you. I hope that helps.
I think that's an important point there, Anastasia, that you raise about information from other sources and not, just be reliant on the EBGM score. And when we've been working with the African Union countries, we've been very mindful that the number of reports and the background volume of data may not always lend itself to the statistical approach. I don't know if there's anything you want to comment on there. We did, Claire Davis did cover some of this last week, but the importance of other data sources when considering a signal, not just being reliant on what might be low numbers of cases coming through the yellow card scheme equivalent.
I could take that. So yeah, there's in addition to just looking at the cases we have and that we receive, there are so many other sources of data or sources of evidence that you can use to kind of bolster your basis, your evidence basis, to inform that decision on which action you're going to take. And this can vary from, maybe it's a new drug, but it's a drug that's in a class of medicines that all act in a similar way by a similar mechanism. And therefore you can postulate on this or make a judgment on the basis of this, that, okay, if these drugs are working via the same mechanism of action to bring about that therapeutic effect, then there is the potential for that mechanism to also carry out and bring about that adverse drug effect. So you can use that to kind of gauge a comparison. You can also look at, this might be more helpful for these the countries in this webinar, what other regulatory authorities have done. You can consult their product information for similar products or similar medicines. What warnings do they have on their reference documents for products. We also cooperate with other international regulatory authorities just to share information, and that's very useful. So that might be helpful. What else? There's literature, obviously, scientific literature. You can just do searches online. Also, recently now with the event of more patients are taking to discussing their experiences of medicines online, patient forums, that can also be quite helpful. Obviously, with these, you have to bear in mind that this is not a confirmation of these reactions being associated with the drugs, but they can be useful to stimulate or trigger that further investigation into these safety issues. So social media and stuff like that. If anyone wants to jump in, I might have missed something.
I think that was very comprehensive, Anastasia. Thank you. And I think you kind of touched there a little bit on what may be behind one of the questions here is the need for electronic data monitoring platforms. And we have spoken about active surveillance as also being a tool in the armory, if you like, rather than being wholly reliant on spontaneous reports and scanning literature and so on. But active surveillance is a key tool for us, particularly when we think about COVID, but in other situations as well, we've used active surveillance. So the comment that we receive, or the question that we received from a colleague from Ethiopia, I don't think we have an answer to this, but I think it is an excellent idea that we have access to electronic records surveillance systems, that we can use technologies like SMS or email, as we're doing in the UK, to actively follow up patients and actively solicit information via that follow-up. So thanks for that comment. Catherine, as you were talking, it struck me that you were talking about batch numbers, specifically with with vaccines, and lack of efficacy you also touched upon. I wonder if we also, and Sam and Anastasia, you may want to comment, but we also look at lack of efficacy to identify quality issues with a drug or a vaccine. Lack of efficacy may indicate a quality issue, even a counterfeit issue. I don't know if you want to comment on that, Catherine, around lack of efficacy being an indicator of other things.
Yeah, thanks. Yeah, it comes with it's really, you see something that's not quite right, and then you go and have a look at it. So in your weekly signals, you see something where somebody's reported with so sometimes it's with, like, this, say, a chickenpox vaccine, and then the patient has reported that they've got chickenpox, and then you start to see clusters of these kind of cases coming in, and I think it's going in and looking at the cases, having this, following up with the reporters as well, because they're the ones that have more details, particularly if you've had a cluster of cases, to establish what might be going on. And again, yes, so you would contact the company to look at their data to see whether there's some with a particular batch, because it's with these biological type products, so not just vaccine biologicals, medicine biological products as well. It's every process in the manufacturing is so tightly controlled, and so the slightest deviation could cause something which has an impact going forward. And it may be that it's something that can be seen. So when the vaccine is made up, or the product is made up, it looks a funny color, or there's some kind of precipitate or something like that, then you would ask the company to look into that and work out what's going on. What's difficult is when you get reports like this where, and it doesn't always come in as an ADR report, it might come in through the Defective Medicine Centre, where healthcare professionals notice something a bit odd with the product, but you don't know what this means. You don't know whether it means it's not going to work and provide protection, or cure the patient, or whether it's actually going to cause harm to the patient. So then, while the company's investigating, you have this decision of what you're going to do. Do you need to kind of take the product off the market? Do you need to communicate? And this is quite challenging, because it all happens in quite a short space of time. You've got to be making these decisions, always keeping in mind that obviously it's the health of the public. But yeah, it's quite situational, isn't it? So you go in and you investigate, and then you take steps kind of based on what you find but it basically involves connecting with the company and also with the public health bodies to try and work out what's going on and what you're going to do. I don't know whether Sam and Anastasia wanted to kind of add to that at all.
No, I think you've
covered it pretty well.
The question came in from Kerrigan McCarthy around, and this I think is very relevant to the sodium valproate issue that we've had, Catherine, around, do we pass on or refer cases on for investigation leading to disciplinary action when there may be medication error, misprescribing, etc., that may be have medico-legal implications. This is one that we've kind of battled for a while in the UK around medication error and the reporting of these. And when there is a specific system for reporting medication error that is done. It was originally anonymous, but anonymous reporting is very hard to get follow-up information. But there were guarantees given that the reports that were made through that system would have no recourse. People should be able to report without fear that a medication error has occurred. Obviously, there are other factors like never events. There's a list of certain kinds of events that should never happen, and there should be measures in place to protect both the system and the patient from these kinds of events. But I don't know, Catherine, if you want to comment on some of the things that have been done around that area, and maybe thinking about sodium valproate in the use in pregnancy, how we kind of mitigate against the use, and how we rely on the sort of medical community to try and reduce the exposure.
Yeah, so as you implied, Mick, our primary function is to so we want people to report adverse drug reactions to us, whether this is a medication error or something, an adverse drug reaction which has occurred under normal use. So we want to collect this data so we can look at the safety of the product. Our role isn't to kind of go back and discipline individuals or pass on details of individuals to, the medical councils, professional bodies in the UK. However, I suppose indirectly in our work that we've done, particularly with sodium valproate, because this is a well-known teratogen. Women with epilepsy need treatment. They can't necessarily, it's not a case of stopping a treatment if you want to get pregnant. But then this is a conversation. It's putting the information out there. So we do the safety analysis to say, look, this is what happens if you take this drug during pregnancy. So you shouldn't get pregnant while you're on this drug. And there are, these are the things that need to be done. So communicating this information to healthcare professionals, providing training materials, and then that in turn, that hopefully kind of stops the process from happening. If another organisation then wants to go and, target individuals who may not have been using the product correctly, that's for them to do. But if we, using the information which is reported to us, we can change or enhance the materials and the methods and the tools that are available to minimize the risk of fetal exposure to teratogens like valproic acid, with things like pregnancy prevention programs. So the patient needs to have the negative pregnancy test, and they need to be very informed of all the risks, because this was the whole review of the valproate issue revealed that in the past there just wasn't, the women just weren't aware of the risks. So if you're not aware of a risk, then you can't make an informed decision. And then it's a kind of cyclical process. So hopefully what we're doing, although we're not kind of disciplining individuals, we are helping the process to, helping the risk not to happen by providing these materials and evaluating the effectiveness of them as well. This is something which is an ongoing thing. We're looking at CPRD data and studies going forward to see how these materials are being used, to see whether they're any good, because if they're not being used or if they're not effective, then they need to be changed. Sorry, I probably went off on a bit of a tangent there.
Very good, thank you. Got another question from Kerrigan McCarthy around the benefits of combining the processes for drugs and vaccines. So saying that in South Africa, there's a separate evaluation, separate reporting channels, etc. for vaccines and drugs. And what are the strengths of combining the two processes? If we take the yellow card scheme as an example, in the UK, we collect everything through the yellow card system, whether it's drug, a reaction, electronic cigarette, herbal medicine, suspected defect reports, etc., etc. They're all called yellow cards. And I think the branding of a reporting system sort of breeds familiarity, and people know where to go to report an adverse incident in the UK, regardless of the type of healthcare product they're reporting on, or the type of event. So a device, a medicine, a vaccine, all through yellow cards. And I think that's borne out with what we've seen with the COVID vaccine. I think it was Anastasia, you said we get around 1,000 reports a week. Well, in the last couple of months, we've been getting 1,000 to 2,000 reports a day because of the volume of reports coming through the yellow card system on the COVID vaccines. So combining that and not having separate reporting channels, I think, has been very important. But there are differences that have been highlighted on how we detect signals, comparing vaccines with vaccines and drugs with drugs. But I don't know, Sam or Anastasia, if you want to talk about the benefits of having joint signal meetings and the sort of experience that's in the room, if you like, and also Catherine, where we use our pharmacovigilance expert advisory group for vaccines and drugs routinely, but there have been special considerations and expert working groups, particularly for the COVID vaccines. So we kind of do have a combined approach, but we then not have a separation of responsibilities as well. So I don't know, Sam or Anastasia, if you want to talk about the signal meeting first, and then Catherine on the expert advice.
Sure. In terms of the processes, yes, we combine the discussion in our standard signal detection meetings. We combine the discussion of vaccines and drugs or medicines together. I guess, like Mick said, just to have all that, knowledge in one room, and people can learn from, previous, safety issues that have gone on. We do separate though, and as we touched upon in our presentation, we subgroup the different types. So we have a different subgroup for vaccines and a different subgroup for just standard medicines. And this enables us to calibrate our sensitivities in terms of picking up certain adverse drug reactions that might be more likely to happen with vaccines over like standard medicines, or to maybe remove any reactions that we usually would expect to see with vaccines, like injection site reactions, arm pain, stuff that we know and we understand is expected to see. And that's where we are fortunate enough to have these statistical programs to implement those automatic subgrouping and give us that context of what's expected and what's not. And especially in terms of COVID now, with our COVID meetings, this is a separate meeting, but we are analysing the number of reports in the context of both different subgroups. So in terms of the vaccine only background and things that you would expect to happen with vaccines only, and then in context of a background of vaccines combined with the number of reports you're getting as well for just normal medicines. And this is useful for us because it flags up different things based on which background and which subgroup we're using, which sort of viewpoint we're using. So the vaccine only background will suppress a lot of those typical local reactions that we see with vaccines, such as injection site reactions. And so a signal for these in the vaccine background will indicate an excess for this vaccine over other vaccines in our data set. So it's comparing the number of reactions we get compared with other vaccines in that one vaccine data set. But then when we look at the combined vaccine data set with normal medicines, we might get a signal more strongly for those local reactions, as the majority of our reports are just for oral medications. So you wouldn't expect to get injection site reactions with those medications. And the advantage of this combined background run is so that we can detect signals for serious ADRs, such as the Guillain-Barré syndrome, that might be suppressed in that vaccine-only background, as this condition is typically reported for vaccines only and not drugs. So in terms of what we're doing now for COVID, we are focusing mainly on that vaccine-only background for signalling, but we keep it in the context of the signals and the numbers of cases flagged by the combined background, just to check for anything serious that might be missed by that vaccine-only subgroup. So again, it's, we don't just silo one. We're always using that context and background of what's happening in both subgroups to use that evidence basis and for consideration before we make any decisions. Catherine, you might want to add something on the EBGEM or the statistical stuff or the pharmacoepidemiology side. I don't know, but yeah.
Thanks, Sam. I actually got sidetracked into reading the comments and can't remember what Mick was asking me to comment on in terms of the different approach. What was it, Mick, specifically?
So just the benefits of having a combined expert advisory group, and then why we might then sort of diverge from that in a specific scenario, like we've done with COVID and having a separate expert working group.
Yeah, I think generally speaking, we have our most used expert advisory group is our Pharmacovigilance expert advisory group, and we have experts. The experts are carefully selected to give us a spread across different, so general practice, pediatrics, experts in different areas of disease. So we've got a rounded, so we can take to them anything on pharmacovigilance. Otherwise, if you're splitting it off on a regular basis, that means you need more people, and it's not quite as streamlined, I suppose. And that it suits us quite well for most of the time. But with the COVID situation and the vaccines, as I mentioned, because it's so fast evolving and it is quite specific, it's very focused on vaccines. And because there are a lot of issues going, you wouldn't want it to, push out of the way any other, because people are still taking other medicines, of course. Mm-hmm. So there are still other, pharmacovigilance needs to continue for non-COVID products. So that's been where it's really good having that specific group on hand, just to give the, the really specific vaccine advice. And then, there's still the standard pharmacovigilance group.
I think that's a really important point. The world's kind of concentrated on COVID vaccines at the moment, but it is really important to recognise that life goes on with regards to pharmacovigilance of all other products. We don't relinquish our responsibilities for everything else just because we're really focused on COVID at the moment. So thanks very much. I am afraid we are out of time. We still have a few questions in the Q&A box that we haven't got to, but we will get to them by correspondence. We always follow up these sessions with a written report from the Q&A box. So the team will collate these questions, we'll respond in writing and make those responses available. So apologies if we haven't got to your particular question, but we will answer. As always with these sessions, we'll now turn to the poll. We'd really appreciate if you can answer the 4 or 5 questions that will appear now, because that will help us for future sessions. And whilst you're doing that, final thank you to the team for your input and your presentations today, and wish everyone a good day. Thank you.
Thanks. You too.
Thank you. Bye, everyone.
Thank you.